Great. Well, I want to welcome everybody to the last of the seminars for this year. We're really excited that we have Blanca Himes here today. I'm going to tell you a little bit about her. She obtained a PhD in medical physics and bioinformatics from the Division of Health Sciences and Tech at MIT. And she was a biomedical informatics research fellow at Harvard Medical School in the computational health informatics program. After serving as a faculty member at Brigham and Women's Hospital and Harvard Med, she moved to UPenn in 2014, where she is now a tenured associate professor of informatics since 2020. Her research focuses on gaining insights into pathogenesis and treatment of asthma and other pulmonary disease using things like electronic health records and multi-omics data. At Penn, she's the director of the Exposure Biology Informatics Core of the Center of Excellence in Environmental Toxicology. And also, I will mention, is a recipient of one of the GG pilot grants. So I'm really excited to hear about the work that you're. I'll let you take it from here.
Okay, thanks. So yes, hopefully you guys are willing to hear a lot of stuff that is not about genes, but of course, all of my life started doing asthma genetics. I think I've worked in the asthma genetics sort of space for 15 years now. Although these days, of course, how we do genetics, as we all know, has changed a bit from like sort of the traditional do GWAS and repeat many, many times. That was maybe happening 10 years ago or more. So today I'm going to tell you about characterizing gene by environment interactions on asthma disparities among adults in Philadelphia. Although we won't quite get to that gene environment stuff, but you'll see why in a little bit. First, I was going to tell you guys about asthma because I know this is not a venue where asthma is discussed often, although I think there was at least one talk this year that focused. So it is a chronic inflammatory lung disease characterized by episodes of airway obstruction where people feel like they cannot breathe. There's no cure for asthma. It's usually managed with treatment of treatment with medications or avoidance of things that cause people to have this airway obstruction. And the things that make people have airway obstruction differ. So in general, you might tell people stay away from them, and that is to avoid those episodes of so-called exacerbation, which usually require people to go to the hospital and get systemic steroids. And so it's uncomfortable. It could result in death, although death is not common in asthma. But that's sort of what we're going for, is disease management with drugs and avoidance as opposed to anything curative. Well-known disparities by race and ethnicity, as well as by socioeconomic status. And here I have some figures from the US from two particular surveys. And I know this is sort of glossing over a lot of groups in the US, but this is how this data is reported. And also notice that I'm giving you guys figures for adults. So people also study childhood asthma, and there are some differences in how these occur and some of their other demographics in addition to their clinical characteristics. So I'm going to talk more today about adult asthma with a few exceptions. So among people who are white, or if you lump all of the Hispanic people together, asthma prevalence tends to be lower than it is in Black adults in the US, although we're not going to get into it in this talk because I'm not going to talk much about Hispanics. The highest prevalence in the US is actually among Puerto Ricans, and one of the lowest is among people of Mexican ancestry or people who identify as Mexican American. So this Hispanic average, you'll see varying numbers, and it usually depends on who the group of Hispanics in the US that you're considering there is. But some of the prevalence in Puerto Rico has been estimated to be as high as 32%. So quite common there. In children, there's not much disparities by gender, although maybe boys have a bit more asthma than girls, but in adulthood, asthma is a lot more prevalent in women. And then you can see here that as the ratio of family income to poverty threshold rises, the asthma prevalence goes down. So those who live in poverty have more asthma than those who do not. In terms of mortality, there's even more striking disparities. So you can see that the mortality among those who are Black in the US is quite high compared to other groups. And it's also more deadly in women. Although the mortality overall is small, as you can see there, those are rates per million, it still is a very striking disparity. So there's a lot of efforts, many types of science to try to figure out why this is. And because asthma is influenced by many factors, there are many candidates that might contribute to disparity. So of course, there's genetics or biological factors like sex and environment, exposures and lifestyle, and then healthcare access and treatment. And this is partly because how we say who has asthma has to do with who goes to seek for treatment and who has the ability to take medicines, right? And so you might capture and say that asthma is more prevalent in some groups simply because people are seen more in hospitals or have different access. First, I'm going to tell you about genetics, especially, not really biological factors. So as for many complex diseases, there are many loci that have been identified for asthma, at least 100 over the past 15 plus years. The first asthma locus that was identified by a GWAS is this very notable one that everybody who studies asthma knows about. It's in the 17q21. They call it this locus, even though this original paper was a very humble one with about 2,300 people. That was sort of the sample size of those original GWAS back then. Now we're in GWAS into millions of people. But despite the fact of having a relatively small sample size, they found a pretty strong peak, and this peak has been observed repeatedly in many GWAS across the world. And different genes may be associated with it and so on. But anyway, we don't need to study too much, or we're not going to go into too much detail on any single gene, but just know that this is maybe like the characteristic one that started it all. Here's a more recent analysis. This is just unpublished using UK Biobank and all of us data from our lab. This is wider European ancestry from both of those cohorts. So now we're talking many more cases and controls than there were in those original GWAS. The signals, of course, lead to much smaller p-values as we see for complex diseases, and we're not going to belabor the point of all of these loci. This is that 17q21 loci that I was just talking about. But one of the ones that has usually very small p-values is the HLA region. So I'm sure many of you know that that's a locus that's well known to be associated with immune responses. It's a highly variable locus. It's actually a pretty tricky one to study.
But can I ask you,
Yeah.
what is known about that or MDL3? Like, it seems like these are all immune related. Is that also known to be?
No. So yeah, I took out those slides, but it's a great question. And there have been there's actually 4 genes in the region. There has been this, some people think it's ORMDL, just DMB, and there's sort of different evidence for each, but they are the subject of many R01 studies. But yes, I had a colleague who was studying, so I'm on papers related to GSDMB, and it has to do with pyroptosis. So cell death, maybe in epithelial cells, I believe, in the lung. That maybe there's some contribution based on exposures, but people have tried mouse models of them, and there's increased RSV infectivity, and RSV is something that frequently is associated with asthma exacerbations. So long story short is there 15 years later, we don't know. There's still a lot of studies, but and as opposed to other conditions where you find a novel locus and maybe it's like a drug target or there's some action out of it, that has not had for this particular peak. And because the other sites that have more to do with cytokine response and things that maybe we would expect more with asthma, especially TSLP was already known before it became a GWAS loci, like the gene itself was known to be associated with asthma. I'd say this is one of the most novel ones, but yeah, in terms of like how you would target it or something for a study. But yeah, in the world of asthma, the people who don't like asthma genetics so much sometimes complain about that, right? Like, where's your drug? Where's your actionable insight that
they can't help it? They told you that.
Well, not me. They just told
Anybody.
the whole community of genetics. But yes,
only PSK9 or whatever.
Yeah,
PSK, like one of the few.
That's right.
That wasn't even a GWAS.
That was from no, it wasn't GWAS, was it?
Rare variants.
Linkage, yeah.
Yeah, or rare variants.
There aren't many success stories, so.
But there may be still, and
That's
you know,
true.
there's plenty of patents around different things as far not you can't take out the patent on a gene, but you know, we'll try to think of what they might do in the future with them. But asthma is certainly, I mean, I just told you asthma has a lot of disparities, and asthma is one of the first diseases where there were asthma researchers that were very early to say, like, if asthma is more prevalent in people who are not of European origin or who identify as white, then shouldn't we be doing GWAS in people of those groups? So many people in the asthma community started to try to gather these cohorts, but nonetheless, there are still relatively fewer GWAS. And of course, if we use GWAS of convenience UK Biobank, then there's going to be less representation of people of diverse ancestry. But here I just have an example of a relatively recent study by Hawk and others, where they tried to pool 3 top cohorts with this other large consortium called Kappa. And Kappa is one of the larger efforts here in the US. It spans a few. Yeah.
Sorry, I didn't mean to interrupt.
No, it's okay.
Wait. So
Like-
does the chromosome 17?
It's not showing up in this
Not
particular.
Yeah, that's what I was.
Is that because the variant is not there? Or?
So it's funny you should ask, because in other GWAS that were smaller sample size of people with African ancestry, then that a locus was there, but then the question is still valid.
Sure.
Like, we don't know. And the other thing is that you know how I was saying adults or asthma in adults and children is different. So that 17q21 locus appears to be a childhood asthma locus. And so if you do a GWAS in adults, the signal usually goes down or is not present.
But this was children, right? It's CHOP.
Yes, this is CHOP, so it should show up. I agreed.
But like, you have more than, I guess there's like 5
Even
times
more,
size. So you
yes.
should have the
So
same
it should be
Yeah.
there. If anything, it shouldn't go away. But yes, that was one of the notice.
The HLA, I don't see that.
Yes, and yes, so thank you. That was the punchline. That this is what they found, even though they concluded still that many of these say that are, the sample size, this is the largest one to date. It has 19,000 participants, not people with asthma. So relatively speaking, the sample size is still small. But you're right that if the 2,000 people GWAS in the UK originally found that GSDMB, then if it's really highly associated, I'd expect to find it here. With childhood asthma and people of all race ethnicity, then maybe you would expect to find it here. But other studies prior to this one have suggested that that locus is there for, I think in the Puerto Rican GWAS it was found, and in the meta-analysis across it's found, which doesn't mean it is there strongly in the individual populations. But yeah, anyway, so CAPA is a very large consortium, and it includes people from different parts of the US, but also the Caribbean, and they have some extended, anyway, it's a complicated set of populations. Some of them are extended pedigrees. And so there is more, but then it's a usual thing, and the same will happen if you Hispanic populations, especially if you start to count that some of them you split people who are from Puerto Rican origin, from Mexican origins. It's a really prevalence of it. There's going to be differences in their genetic association as well. Maybe
I
not.
thought, was that the disorder where they showed in the Puerto Ricans that depending on the percent ancestry or local ancestry, African, Native American,
Yes. So
European, that there was, and then also with wealth, right? I thought there was also wealth was like a cofactor or something.
they probably
and-
tried to put that in there. Yes, but so that's right. Those were a step on.
Yeah.
So that's right. Because that was part of the question of like why, and there's others. So there's other Puerto. Sorry, I'm like, we're not going to talk about Hispanics. No, let's talk about it. But yeah, so then the question is like, is it Puerto Rico, the island? So maybe is it because some people study the Saharan dust that comes across, or is there some other local factor? But no, Puerto Ricans, there's a Hartford, Connecticut cohort as well, and asthma is highly prevalent there, suggesting that maybe it's not the environment. So yeah, one of the hypotheses was that Hispanic populations have different amount of admixture from 3 continents. So maybe that was what it had to do with that. Maybe it was the fact that Puerto Ricans are more similar to people of African ancestry than not, because there is a lot more admixture with people from Africa. So yeah, they tried to quantify that. I don't know how well they did, but on a locus by locus basis, that's not shown. But yeah, that is one of the hypotheses. If you're going to talk about asthma genetics, then maybe that's why the prevalence is higher. But if you talk to any health services researcher, they will tell you like, no, it's not about genetics. It's about these other things. So we can be simplistic, but it probably has to do with all of these factors sort of coming together, right? Like none of these genetic loci are deterministic at all. So maybe there's sort of like this complicated sort of factors coming together. But nonetheless, even if you're doing a GWAS, especially if you're doing it in a biobank, like you can't ignore the role of the environment, exposures, and lifestyle, or we can ignore it because we oftentimes don't know how to quantify it. Like we don't have good measures of it, especially when it may have mattered, which is studying adults may have mattered what they were exposed to in childhood and so on. Healthcare access and treatment to some extent, maybe you can use some of this information or extract it from one of these data sources. So now that segues into electronic health records. What I showed you earlier mostly had to do with genetic epi studies, except for of course, all of us in the UK Biobank. But electronic health records are going to be valuable for a lot of this biobank-based research, although here I'm just talking about them more broadly. They just provide convenient and low-cost cost access to longitudinal information for many people, more than you would be able to enroll in most studies. So the easiest thing you can do with them is just call people. And then once you enroll them in a study, you can really get the equivalent that you would get in an epidemiologic study, right? Which is just comprehensive information for every participant gathered the same way. You might just use it at the point of care, which is not what we're talking about now, right? But of course, there's the type of informatics where you go into the hospital to prove things for their delivery. For biobanks, of course, it's kind of how a lot of biobanks start these days, just because of convenience. And the derived data could be used for primary research, which is one of the things I try to do as well. And of course, some people, more skeptical people would think immediately that this shouldn't be done. And of course, when I was a grad student, everybody, especially in epi, thought it was crazy to even create a biobank. But nowadays, like almost every health system has them, right? Or every large research system has them. It's something that we sort of take for granted that we can and we should create biobanks. But nonetheless, important health-related information is missing from the EHRs, something that we don't forget just because we're using EHR data. And that has to do with variables related to economic status, education and health literacy, and environmental exposures. And for a condition like asthma, all of these would matter a lot, as we just see from just looking at epidemiology of the of who has asthma. So we've tried in various ways to try to expand the scope of EHR data, and research, like sort of recent efforts, have to do with including things via geographic information, because that is a cost-effective way to do it. So we might use patient geocodes in different ways to link such data sets to electronic health record data. For example, by people's residence, but in children, maybe it could be by their school or something else. For adults, it might have to do with their place of work. And we can link external variables and include them as covariate. So we can try to estimate the measure for each person and sort of just use that in a plain regression model. Or we can try to do spatial analysis depending on the coverage that we have. But again, the idea is to take some kind of geographic information from the electronic health record and then bring on these other layers of environmental or social information. And this is also possibly, of course, because there's so much publicly available data, at least here in the US, right? Depending on the different country, or if you're trying to use global data, your mileage may vary. And of course, the data quality varies based on the source of the data. Some data is not as accurate as others, the resolution relative to the needs that you have, the amount of missingness. And so again, it's not that any particular data set is terrible or amazing. It usually has to do with, like, is it appropriate for the purposes of the study design that you have and the question that you're trying to address. So let's talk a little bit about Penn Medicine asthma patients. And this is not the biobank. This is just looking at our data. The earlier efforts when I arrived at Penn that we looked at is, let's just see who has asthma as defined by these ICD codes, which are billing codes. Not necessarily the most accurate way to classify diseases, but it's very commonly used in biobanks. And the other problem, of course, if you just look at electronic health record data, is that who is your comparator group? So we can see that 75% of the patients were female. So it sounds like a lot more women, consistent with what we know about the epidemiology of asthma. And we have about half of the patients were white and Black. And really both of these groups, this is non-Hispanic, make up about 92% of the records, at least at that time. So the other groups are also present there, but just for proportions, which is why I'm only showing these sort of broad classification groups. But if we don't have a reference group, we wouldn't know, like, what does this mean? Is this a disparity or not? Because who is sort of like our underlying population controlling for? We don't have a good answer for that because there's bias in who comes to the health system. But maybe one thing we could do is just compare those proportions to the proportion of all patients who visit Penn Medicine. So for that same time period, there were 3.2 or so million patients who came through, and of all the patients, 60% were female. So if you just did a chi-square test, or if you just look at this, like, okay, we're enriched for female patients with asthma, and all patients who saw the health system, 29% were Black. So at least by some coarse metric, we would say, yes, there is an overrepresentation of patients who are Black who have asthma, which is also consistent with national epidemiology observations. And if we focus only on those who have asthma, then we can try to measure things like who has more exacerbations, and there we might be able to see the disparities more clearly, because now we're not going to rely on some control group of people who don't have asthma. And so here, using a simple definition for exacerbations, which is consistent with clinical guidelines, we're going to say, let's see who has had a prescription for oral steroids with a primary asthma diagnosis during their encounter. And we're just splitting people into groups. So number of subjects, most people in this given time period had no exacerbations as by that definition. 30% had 1 to 2, and fewer had 3 to 4 and 5+. 5+ would have been a lot of so-called exacerbations in this time period. If we look at the proportion of female patients, they're pretty stable across all of these groups. But if you look at the proportion of Black, so Black versus white, remember I've excluded all the other groups. Now you start to see a very striking disparity. That is, you get to the number of more exacerbations. We're talking like 90%, 5+. It's a small group, but that still is a lot of people with exacerbations. So market disparities, certainly in exacerbations, even though it's harder to quantify the disparities prevalence. So if we're sticking to the EHR data, I know this one's, don't strain your eyes. We can try to do the classic thing where we take as many variables from the electronic health records as we can, BMI, smoking, comorbidities. We see things that we might expect, like chronic bronchitis is a high risk factor if we're also having exacerbations, which just, you know, you have a similar lung disease or potentially classified one that just means you have more episodes. So there's so much we can get from just using the EHR data. Another thing that we might do is, again, take the geocodes of where people reside and try to understand spatial distributions. In this case, we're going to use residential address, and we can perform, instead of a regression model, a spatial model. Here's just a simple one that is in an R package called MapGam, for those of you that. So it's a generalized additive model where you add in a special term for a smoothing term with latitude and longitude, and then you do a permutation analysis to get p-values or quantify your significance. So you get a global spatial heterogeneity is present if you see that people are not distributed as you would expect by chance, and you might identify hotspots or cold spots with contour lines. So one of the first times that we tried to do this, way back in the day, we found this map. Of course, most people who see Penn Medicine are closer. So these days we would not try to get such a large geographic area, but pretty much anything that we do, we usually see a hotspot in West Philly towards South Philly, no matter how many times we repeat the definitions and so on. So the question is, like, what do these spatial distributions mean? And of course, Philadelphia is a city with diverse neighborhoods and environmental features. We also have a lot of segregation, right? For those of us who live in Philadelphia, we have noticed that, and for those too, our neighborhoods historically are quite segregated. And what we would say in terms of environment is that Philadelphia has many environmental justice areas. This is a term that's used nationally, but there is like a state definition that actually changed recently, but this was a definition until 2023. An environmental justice area is a census tract where 20% or more people live in poverty and 30% or more minority population. So by that definition, almost all of the land mass of Philadelphia qualifies as an environmental justice area. And other parts of our state and the country do as well. But that is important because these residents in these areas are exposed to greater levels of emission from trash incinerators, vehicular traffic, oil refineries, even though one of our largest oil refineries is no longer there. But here in Philadelphia, there's plenty of sources of pollution that disproportionately affect some people that live near to them. And some parts in environmental justice areas in Philadelphia also experience higher pavement density, reduced tree canopy, limited access to air conditioned spaces, and many other characteristics that might put people at risk. And we already know from the literature that air pollution is one of these many things that contributes to asthma. There's extensive literature. I'm not going to recapitulate it here, but these are the 6 criteria, or 5 of the 6 criteria pollutants from the Clean Air Act have been associated with asthma. TRAP stands for traffic-related air pollution. That has also been widely associated with asthma outcomes, including prevalence, more symptoms, visits, hospitalizations. So it might be important to try to quantify air pollution and link that to asthma if only we could, and if we could do that accurately. And one common way that we try to relate air pollution data to any outcome in the US is we use EPA regulatory monitor data. They have the most comprehensive system across the country. And that's just, these black dots represent the sites of the monitors. And these have been around again since, for quite some time, since to try to enforce the Clean Air Act. And so they measure these criteria pollutants and a few others, but these are the ones that are measured nationally. Of course, you can't use point data to try to infer people's pollution. And so there's a lot of efforts to try to do interpolation, or how do you go from one particular person's location or locations and try to estimate pollution they've been exposed to. So we've been using this data for some time, and we've gotten to the point where we created an app that just has interpolated data so that we can take a geocode and then infer the pollution at that site for various criteria pollutants.
Do you know how many of those monitors are actually in Philly?
Yes. Thank you. The city of Philadelphia, we have this public health air management services, and that is literally part of the grid, even though they're called AMS, or the air management services. They monitor our local air pollution, and I would say they have relatively sparse geographic coverage. It depends on what you're trying to do. And most of the monitors are placed near sites of high traffic volume, because that's one of the things they're trying to measure. I apologize to those of you who are colorblind. This is a plot by Philly Gov, not me, but there are red dots on this screen, and those are the main sites, the main network from at least 2021 regulatory monitors. And I know here's another table where you have to squint to see, but they don't just measure the 6 criteria pollutants. And in fact, there is a monitor that was near the refinery that was trying to measure other pollutants that the refinery was not supposed to emit, at least past certain levels. So there's other purposes, but if I showed you these over where the I-95 corridor and some of the other freeways are, some of them are located right there. So if I were trying to assess, is pollution different in parts of West Philly versus Center City, it would actually be quite hard to do, because there's not sort of monitors there. So more recently, though, there are satellite-based measures of pollution. The other alternative, by the way, that I'm not going to talk about is there's sort of increasing desire to use sort of low-cost pollution sensors, right? So can we all either place low-cost monitors outside of people's homes, inside their homes? Maybe you walk around with one. I'm not going to talk about those here, but another alternative is satellite-based measures of pollution. So Tropomi is one of the instruments that I know. Satellites, we're going all the way to satellites from genetics. But Tropomi is one of the instruments that's from the European Space Agency, and that one measures a lot of things. It's trying to measure climate change, things that different components of the atmosphere that might change in response to climate change. And so Tropomi, the one that we care about, is measurements of NO2, or that's one of the things that we're going to look at. It measures a tropospheric, column density of NO2. And so there's people who have done chemical transport models to try to infer, based on what's in the atmosphere, what might be at the ground level. And the correlation is pretty high, at least at some sites around the world that we tried to verify. Not we, Cooper et al. Tried to verify to make sure that there was sort of consistency with the model, and there was. And more recently, at least for North America, there is this new Tempo satellite. The data is not quite publicly available yet, but this is a satellite that's parked above the Northern Hemisphere. And so Mexico, Canada, and the US will have constant measures at a very high resolution compared to these other instruments. So this will probably be the alternative that everybody starts to use in going forward, at least in North America.
What kind of resolution?
1 by 1 kilometer grid, or even a little bit smaller, and I think it's hourly. So
Tempo, yeah.
So NASA has this data somewhere, and yes, there's an early adopters group right now, but they're trying to figure out how to keep this data in a way that it's useful for a lot of people, because it is such big data. But there's other very large data sets in Google Earth Engine, and some of those can be used. I think actually the Tropomi measures might be in Google Earth Engine, but there's all kinds of other global data based, satellite based mostly. Some of it is not satellites, but LiDAR or other lower Earth devices that could be used for various studies. But we tried to just see okay, well, can we use these measures instead of the EPA regulatory monitor one? So a few years ago, a student was an earth sciences student. She took the measures at 3 of the geocodes for 3 of the sites here in Philadelphia. In fact, these are the sites, not that we need know the name of every one. But there was pretty good correlation. This is just for the monthly measures of NO2. And if you look at the seasonal variation, according to the ground level versus satellite NO2 at each of them, it was, and by ground level, I mean the EPA regulatory monitors, there's pretty good consistency. So at least for those sites, it looks like, you know, we have consistent measures. So maybe the satellite data is accurate enough that we can use it outside of those same geocodes and just trust that it's going to be good at all of the other places. And if we look at the distribution of those different data sets, this is what we would get. So the interpolated EPA data is here. And those are like the few sites in Philadelphia that have measures. And so you can see if you try to do interpolation for these faraway places from the monitors, chances are you're not doing a very good job. But whether you use the raw satellite data or the ground level estimates, maybe based on where our industry is located, it's probably accurate that there's more pollution down here. And maybe the ground level showing that there's more variation across land is even more accurate, although we don't really have a way to verify that one way or another, but it's something that we might consider. That's all I'm going to say about satellite data, and I'm not going to give you as long of a story of each other data set, but just to give you a flavor for other data sets that we've considered using. There's a toxic release inventory, and we were specifically looking at air releases. This is voluntary data that industry has to provide if they meet certain, or if they go beyond certain amounts according to EPA guidelines. But there's toxic release inventory data for other things like water pollution, and so on. The traffic is really important as a source of emissions, and so we can use State Department of Pennsylvania transportation data to look at roadways, at least the major roadways. And with that, we can quantify things like daily vehicle distance traveled, which is a measure of presumably more exposures. And that's why you see the major roadways having higher levels. Area level socioeconomic status. So these are classically used in so many studies these days. The main ones are area deprivation index. And this is a so-called measure of socioeconomic disadvantage. It's actually derived from 17 American Community Survey variables. So that's US Census variables that have to do with like, what's the median income in this place? Is there plumbing? There's a certain, what's the mean level of education? So they tried to make a composite score based on those variables and then quantify it. And this measure is available at the census block level. And in fact, the people who make it recommend you use it at that particular level, which is pretty small unit. And then there's another measure called the Social Vulnerability Index. This was made for a different purpose. These are supposed to be areas with limited ability to prevent human suffering and financial loss in the event of a disaster. Although it's also based on a composite of American Community Survey variables and I think a few other things. This one, they recommend that you use it at the tract level, so at a larger geographic area than ADI. And if you look at these two maps in Philadelphia, you would say, well, they kind of look correlated. Can we use them interchangeably? And I think some people in some papers use one or the other or both. But like they're actually supposed to be different. And if you look at them at a national level, you can actually see not really correlated nationally. Area deprivation index to me looks more like the places where people have high income and the high housing prices have less deprivation. Whereas social vulnerability index, it looks like it does take into account maybe somehow who has more weather events or something, which is kind of what it was intended to measure. So just keep that in mind that if you see one or the other of these used, it may or may not be appropriate. Maybe using the census variable itself would be better. Maybe just using the mean prices of homes is a good marker. That's sort of up. I don't think there's consensus out there on the ideal one. Exposures related to built environment, things like housing code violations. Lots can be said about that. Normalized difference vegetation index, that's a commonly used one, but again, a lot to be said about that. Some people like that measure, some don't. All of these measures, of course, can be correlated with each other and with many other things. But nonetheless, we have a lot of these possible measures that we can use to understand various health outcomes, including asthma. So let's say if we're going to try to take the spatial analysis data sets, how can we do that? And here I'm just going to give you a flavor of it. So we're going to try to select asthma patients with like more stringent criteria to try to make sure that we have a more so that if we see exacerbations, they maybe have more to do with something similar and not some kind of bias. We're still going to try to adjust for all of the EHR data and then maybe we see what happens. So we're looking here at exacerbations. Anybody with 10 or more exacerbations was excluded under the presumption that they're maybe just taking oral steroids. And we're also going to apply a spatial filter. So that's one thing that we don't have going for us in our areas, that there's a lot of competing health providers. In some parts of the country, if you say these are all the people who have seen a hospital, or in some countries, right, you really have a catalog of all of the encounters. But in our area, there are many competing providers. And so we don't have that luxury of like, maybe the patients come here sometimes to Penn Medicine, but maybe on another day they go to Temple or to Jefferson or to any other number of places. And so one thing that we can do to try to mitigate that bias at least a little bit is to try to find a spatial representation ratio where we just see what's the fair health system based on the people who live in the given census tract. And from that, we get this ratio, and then maybe we're going to filter out zip codes of people who have a pretty low spatial representation ratio, and maybe that will help correct this bias. And it's a principled way basically to get rid of those zones that we just think that if people have asthma, they're probably not going to come and see a Penn Medicine provider. They might just go to another local place. And then let's perform our generalized additive models again. So the univariate model for that region again shows this really high peak in sort of West Philly that extends down into South Philly. And if we try to adjust for all the EHR variables, we do see that our odds ratios decrease a lot, but there still is a significant site. And so that is part of our ongoing work, is to figure out how to take all of those other data sets in some principled way and integrate them to see if we can help account for the variability that's still present. And with that, I will start talking about the PBB, which was the topic of how, you know, we're so far I haven't even talked about Penn genetics data. We're just focused on EHR data itself. But PMBB is based on our EHR data. It's not based on anything else, at least at the moment. So can we try to incorporate some of these social and pollution variables with PMBB to study asthma, and potentially anybody else can try to use them for their own disease of interest? So let's first talk about what we have with our genotype data. I'm not going to tell you guys about Penn Medicine Biobank. Some of you guys who are brand new students to Penn have not heard about the Penn Medicine Biobank, but it's a resource here at Penn where they actually do have consent from about 250,000 people to do genetic studies, so which sounds pretty impressive. That is a pretty large sample size, but in reality, there's currently genetic data on about 42,000 people, and there's sort of two rounds of data that have been generated. So exome sequencing data is the most recent one, and there's about 43,731 samples with variants after we perform various filters. This is people of all genetic ancestry included together. And then there's older genotype microarray data with imputed variants, and sort of using traditional GWAS approaches, you can analyze each of those.
So presumably they're overlapping.
So when they redid the exome sequence, yes, they're the same people for the most part, yeah. Because when they redid the exome sequencing data, well, there was the 12,000, yes. So forget it, yes, these are overlapping. They're not like different sets of people. Yes, and they update the phenotype data once in a while, but the phenotype is most based on ICD codes and some other lab values. But what you can do is that you can write an IRB and say that you want to link more information to the Penn Medicine Biobank, and then an honest broker can do that. And so in our case, let's say if we have EHR data on, I don't know, 80,000 people, then we can try to see of those 80,000 people, how many of them are in the Penn Medicine Biobank, and then presumably have more detailed phenotype information, just because we spent a lot of time cleaning it to do more. So if we perform an asthma GWAS, then that means we're going to try to find out who has asthma, and we're going to do that very simple billing codes. We're going to rely on the genetic ancestry computed by PMBB staff, because that's what one does if one uses the PMBB. Although you can try to maybe explain to them why you would need to do something different, but in general, that's how one does it. Because the vast majority of participants were identified as white or Black/African American, then they themselves tried to do this comparison to 1,000 Genomes Project, and a lot of the participants end up in one of those two groups. That is the labels that PMB gave them to say that that's their genetic ancestry, although I feel like the African especially are very admixed. And then we just do a pretty vanilla association analysis. These days, one does not have to reinvent the wheel to us. And if we do that, we're going to use sort of traditional thresholds for counting something as genome-wide significant. And actually, if we do that, we still get no genome-wide significant findings. However, at least if we use all participants, so the number of cases with asthma would be about 7,200, and we'd have about 28,000 controls. Our controls for the most part are everyone else, but we did try to filter out for people who had codes for COPD or a few other conditions. And our top SNP still is in the HLA locus. So that gives us some hope that maybe there is signal there, even though find significance. But a lot of those other findings that we might expect are not present. We did things like check if this association had anything to do with people with autoimmune diseases that might also have HLA variant associations, and that is not it does not seem to be due to that. So it does seem like it's an asthma locus. But keeping in mind, this is all participants. We did adjust for ancestry, but if we try to just do a stratified analysis where we look at each ancestral group separately and we it seems like those results are mostly driven by the ones with European ancestry. And if we look at the people that end up in the African ancestry population, then we don't even see like maybe even suggestive signals, not much of anything. And so here I'm just trying to show in a table, again, not to make you guys squint too much, that I think this, if the GWAS are in all participants, most of them are being driven by p-values in the European populations, although at least the direction of effect is pretty consistent. And that's why when you go and do the meta-analysis of both, you still see these showing up in all participants. So HLA region is there, which could be a true signal based on what we know about GWAS of asthma conducted in populations. So, oh, and I forgot to mention that I'm just showing you the exome data, the exome sequencing data. If we do the GWAS using the imputed data based on the genotyping microarray, now we have like a little bit more suggestive peaks, still nothing that's genome-wide significance. HLA region is still one of the top loci, and at least in the one of everybody combined, there's a few other loci that would show up in a high-powered GWAS, like IL-1 receptor or L1. But if we split this up again by ancestry, we just have very few loci. And they actually do look quite different this time. If we do us versus the exome sequencing, we discover more of the gene in all of those pieces. You know, again, some of the sites that we expect to be there are there, but it's hard to know like if the rest of it is just like a scrambled mess or if any of those could represent a true signal until we have more participants. Okay, so that's starting with the GWAS part of Penn Medicine Biobank, and then we're trying to link this external data. So what happens if we try to do that? So we had to work with the PMBB to do this because as honest brokers, they don't want to share the geocodes. Geocodes, of course, are protected health information because, you know, if you kind of know a place or a site where somebody lives, then there is a high risk of re-identification if you were not an honest person and tried to re-identify people. But this is where the PMBB people live, all over the place. Who knew?
Yeah, that's a surprise.
So there's very few people in other places, but they're still like, they're kind of representing most of the US. Yeah.
This is where they live now.
Now?
Yeah. Like,
Or
or as of 6 months ago. Yes, not when they were recruited. Sadly, our EHR does not keep the history of that. Oh my gosh, yes. Geocoding in our EHR is a whole nother question,
So
but yeah.
Is this mostly like people having moved in the last like 10 years?
We couldn't
basically?
tell you
Like,
for
moved
sure.
away from Philadelphia.
That's my guess, but I couldn't tell you for sure because I don't have data. Well,
Presumably these people weren't actually
visiting
living
here,
places when they were patients at Penn.
or they were here for a visit and then for like that one visit, or I don't know, some of them have 2 houses. I don't know. There's different scenarios.
How far back would?
Oh my gosh, some of the patients go back quite far because it's genetics. And so you can take the blood draw the one time and then be done. So I think some of Dan Rader's cohorts, I don't even know, do you know, Sarah, when Dan, because the PMBB started with some of Dan Rader's cardiovascular cohorts. And so some of those participants probably go back 15 years. But many of the genotype ones, they're not recent. Like, so even though we're using their recent address, like they donated blood at least, I don't know, 5. When did Regeneron do that second round of sequencing? I feel like they started it 5 years ago, right?
Were they like students or something? And then I could see if they were
Yes.
students and then they left and they got, you know, went back
Yeah,
home
for
or
sure.
got a job. Jobs.
I think these are mostly like people who were in the like CAD clinic.
Which?
like the cardiovascular disease.
Like they came in.
You know,
And a question.
Yeah.
Are these guys for the whole cohort or just the genetic?
This is just this is the 43,000 or so with genetics data currently, not the 2,000. Yeah, for
is this like, I mean, are these like, what's the scale? South Dakota, is that like one person?
no. So these, because I can't show you geocode, so I'm plotting them. So 3-digit zip code is the smallest geographic area that's not considered protected.
Yeah, no, yeah, but I mean, like, does this mean that like there's just one participant who
There
like,
could be,
moved back to South Dakota?
there could be, yeah.
Yeah.
Yeah, so like they're close to not so many.
Like, I mean, yeah.
Yes, and actually I'll show you the zoom in of the area that we care about in a second. But yes, so you're absolutely right that they represent a lot of places, but that doesn't mean exclude them. I'm just saying that sadly, when you don't have a lot of people to begin with, it's a sad thing to say, to throw others out. But we certainly, if we are going to study something like where time matters, like asthma, and if we're going to talk about exposures, then would care to have some proximal window of the exposures, right? And so if they don't currently live in Philadelphia, then it's harder to, anyway, to try to use them for a study that does use that data. So this is looking at our region, and I was still surprised to find out, I guess it's still hard to see, but 30% or so are in Philadelphia proper. So the city or county of Philadelphia is the same thing. But this is again, a 3-digit zip code. So Philadelphia and then some of the surrounding areas, if we start to put them together, we start to get higher and higher. And then by the time we include any of the 3-digit zip codes that are in the 3 states sort of within this window, then we do get a lot of the participants, but I don't know, we're still throwing out a good number in my opinion. But if we are going to think about environmental variables and we're already making so many assumptions, then I would say we certainly can't use the people in Montana and compare them to the in Pennsylvania.
But I guess what I'm wondering then, so these are people, they could have been collected 10 years ago. Let's say you go out, you get the environmental information from today. So I guess presumably you still have health records that are ongoing, is that, because otherwise, what good is it? Like if they were.
Exactly, it may not be. Well, we could go back with the pollution, but then that's a whole nother story, right? Like, do we try to like, because this is the current residence, so we wouldn't know what the residence was back then, right? And so how do we, that linkage? Yes, very tricky. I think the best bet is to stick to the people. The problem is we don't have a way of knowing. Yeah, I guess if they're currently around here or they have been here in the past 5 years, then it's okay that they were living somewhere else 10 years. But not the other way around.
But if people moved like within Philadelphia,
Then they're totally misassigned. Exactly. So if you went, and especially if you went from like, I don't know, living next to the oil refinery to
Yeah.
somewhere else. But yeah, that's where it's a long story how the Penn Medicine did the geocoding, but they used to do it every 6 months, but then they kind of had to stop doing that. And it was my hope that at some point we used like, look to see what the rate of moving was. And so
presumably you care about where they were when they were children.
no, because some people have, in fact, anyway, yes, I left that part out, but yes, no, for the adult asthma, now, like with the exposures now, especially if I'm like,
So childhood exposure doesn't affect adult
it
asthma.
may, but that one I would not have any hope to capture anyway, unless I were studying it in children. You know, like if I were studying CHOP and I were looking at, then I would. But for many adults, the thinking is that they've just had, it's a different type of asthma. So they've just had more of a burden of exposure to pollution and other things that has just like made asthma onset more recent.
Could you start with the EHR and then try to recruit people for the biobank based on where they're living? You know what I mean? Like,
Yeah.
Go out to the people. Would you be interested in participating?
Could be.
There any chance you could look directly at when the oil refinery shut down and if there was a jump or change, everything else?
We have tried to do that, but yes, I don't even have those slides, but yes, we did try to do that and well, we tried to see if there was a decrease after, right?
Sure, right.
But so the interesting thing about that is that using the regulatory monitors, we could not even measure a change in pollution. Which was actually, even during the explosion, that was like a whole nother thing, but the residents that live there who have organizations, like there's a Philly Thrive organization, and they've been complaining about the oil refinery for so long.
Sure.
And then researchers had to explain to them that the sensor from the EPA that's right there, that is mandated to be there to measure oil refinery pollution, didn't capture anything. And so there's explanation. It's because the explosion was so large that it went up high into the atmosphere. The wind blew it away from the direction of the monitor. But the people that are like, what good is that monitor then?
Wait, what explosion?
The oil refinery in 2019, there was a large explosion at like 4 a.m. And that's what finally got that refinery shut down, because I think it went through 3 different vendors. And yes, that's another story. But let me finish this one really quick and then we can chat. So we still did try, in the end, I did have the PM people. We had to go like give them scripts and their basic data to do most of this. Like, of course, the ACS and some like people include them in their studies all the time. And so they have included this to the extent possible. And we just wanted to see like, okay, what is the distribution? And most of these we did do the most recent year available, although of course we can go back as far as we want. It's just like how good will that, or what good will that do? For ADI and SVI, I think there are people who think it's okay to use current one because fewer people move across SVI and ADI. I don't know if that's true, or I've not seen that quantified well. But in any case, here's what the mean ADI. So I'm not showing you the per person, although the resolution for ADI would be at the block level. That's why this map on the left looks a little bit like broken compared to the map on the right, where it's at the tract level, larger geographic area. So here I'm just showing you like the possible values that people can have. So if people all are living in one place, for example, Philadelphia, we wouldn't have differences across their SVI or ADI. Well, we would for the one at the block level. But anyway, so we are assigning them to each person, even though in the maps I'm showing you what they look like at a broad one, because I can't show you in people. So there is some variability, and this is partly why we don't ever want to include only the parts of Philadelphia, because Philadelphia in general tends to have more deprivation than the surrounding suburbs, for example. So for those types of exposure studies, we might want to look at both. And for PM 2.5 and NO2, here's just the mean of the regulatory monitors. So it turns out like if we measure the mean of the regulatory monitors for those areas, there's not that much variability, even though in the map, I'm sort of suggesting that there is based on the coloring scheme that was selected to show variability. But yes, for example, just to give you a sense, during the wildfires last summer, the PM 2.5 went up to like 200, 250, right? So that would be an extreme level of pollution. But it still is this question of, does it make a difference, right? Because we don't know for sure what the best level of pollution is. So maybe this change, it's one that has been associated in prior studies, although it's unclear where that threshold would be of sort of good versus bad exposure. Okay, so those are all the data that we put together. We can augment EHR and PBM data with these external variables to try to understand health risk factors. But as you can see, there's sort of a lot of caveats, and the small sample size of participants with asthma specifically for us makes it really hard to do a G/E study. But we're hoping that at least maybe with some additional covariates, we might be able to improve the GWAS signal. But that one is to be determined as we still figure out which of the data that we've incorporated makes the most sense to use. So we're still sort of exploring the distribution of all of those variables to figure out what makes sense. Okay, so on that note, yes, thank you, Sarah and Global Genomics and Health Equity Center for funding this. Hopefully those variables will help other studies as well. Depending, I think, again, the catch is like, what are you studying, and does something like ADI or SCI matter a lot? And yes, depending on the outcome, it may or may not be a helpful data set to include in a study. And thank you to the many people who especially contributed to some of the slides that I've showed you just now. All right. Oh, hello, online people that I have not talked to yet, but they're welcome to talk to, right?
Oh, well, should we say?
Oh, okay, sorry.
Thank you.
So quickly, if you'd like to.
Yeah, so we still have a few minutes. We've been asking questions all along, but, or if there's someone online, I don't know. Is there's no questions online? Okay, does anybody have more questions?
Thoughts? Oh, yeah, go ahead.
Yeah, so I wanted to find out, with your SVI plots, for example, can you identify, like maybe the individual ID so you can tell which race and what kind of environment?
Yes.
Okay, have you checked, for example, or you haven't started the gene environment?
Correct, yeah. Yeah, just because we're looking at the gene, if there's not much there with the genes, then I think proceeding to the gene by environment, like as a proper statistical analysis, like it's hard enough to do a gene by environment study when you have a large sample size, but with our sample size, what we're thinking is we might just use them as covariates rather than do that properly. But yes, SVI, so that is actually one of the problems that we have just from an analytical standpoint, is that because there's so much segregation in Philadelphia, then it's hard to tease apart the environment from race, right? And so then we're like, well, then what do we do, right? Because then the study would have to be like, do we relocate people, which is not going to happen, right? Although Johns Hopkins did do that anyway, yes, people have tried to do that on a small scale. But yeah, for us, it's like, if we do the stratified analysis within, and then we have an even smaller sample size, but maybe that's more reasonable, right? Because then maybe we're adjusting, like we have a smaller geographic area, but then we also need the variability within that smaller geographic area to be at a high resolution differences.
Okay, and then, so I think there are two other factors, like heritability in your study. How can we?
Oh yeah, so what's the heritability of asthma? That's a great question, and I didn't bring it up. So I think the estimates range from about 0.45 to 0.98, which is a little bit silly. But I think these days people would accept that it's close to 0.6. But I think it depends, right? Because asthma is one of those conditions, like I was mentioning, that maybe childhood asthma is more heritable than adult asthma, and adult asthma may environment. But then within asthma globally, there's a lot of effort right now to try to understand, are there subtypes, or in asthma, we would call them endotypes of asthma. So are there different different immune signals or profiles that people have that maybe give, like some people are really allergic when they have asthma, and other people, it has to do with exercise. For some people, obese asthma is a big thing these days too. So some people never had asthma, and then clinically obese asthma tends not to have a Th2 phenotype. So maybe the heritability of each of those types is also different. And if you just do a biobank and group a whole bunch of people and you get your heritability estimate, that might be a ballpark, but maybe in the future we will be able to fine tune which are the more heritable types.
And then integrations, I mean, some of that come from other countries.
Absolutely. Yeah, we're so limited with oh my gosh, I will not, yes, I did not even bring that up here, but yes, if you want to see a nightmare, try to classify race or ethnicity in a biobank or in an EHR. At least now there's codified data, but in the past, I think there was also like text boxes. And yes, we see some craziness there, but unfortunately, I think without a survey, we won't do better.
But you could use genetically inferred.
Yes, that's what we do now. Correct.
And then also, could you try to estimate polygenic risk scores, let's say from the bigger studies, see if they're effective in the
In these
patients
ones,
here.
yeah.
If so, then you could maybe use that in your, I don't know, see the impact of environment
On those two groups.
considering high risk, genetically high risk for
Yeah, I think there's polygenic risk scores for childhood asthma too, or
Mhm.
yes, those change a little bit, but yeah, no, that's a good idea. That can certainly be a way to try to just use the genetic information in a different way as sort of one big consolidated measure instead of try to find the individual ones to understand.
Yeah, and one last question.
Yeah.
Have you checked PWASs and Mendelian randomization studies to kind of find other rare, it's the ones you're currently using?
Yes, there's so many Mendelian randomization studies, but yes, I have problems of assumptions of them. I would say are like a big barrier to some of these, because some of, yes, there's things like smoking, like we have tried to look at that, right? Even though smoking is classically related to COPD, it's also associated with especially adult asthma. But yeah, and like trying to, I think under a lot of those assumptions, by the time you bring in other variables, right, like to really set each one completely independent becomes difficult. But yes, there are many other variables, and there are also like I didn't show national data sets, but there are some in the US about healthcare access, distance you have to travel to get to your provider. A lot of the providers here would tell me that that's a large barrier for patients, right? Like some of them, it takes them 2 hours to get to the doctor, or a really long time to get to the pharmacy. So there's variables, many other things that we could think about incorporating.
Yeah, like, but I think like that.
Oh my gosh, yes, diet. That's going to be hard without personal data too. But yeah, for a while, and they're still out there, right? Was it the FDA that put out the food deserts? it's such a coarse measure, right? But it is, but still, anyway. Food deserts, I think, is maybe the only area level way to say that there is, except that food deserts, guess what they're correlated with? These other areas. So I don't know if you're adding that much more by separately including some a food desert. I think at that point you would want to get dietary information from individual people.
Any other questions? And if not, we're already past the hour, so thank you so much. It was super interesting.